Okay, let's start our last session of today. We have Professor Jacob. He's an associate professor at the Department of Mathematics and Computer Science at Eindhoven University of Technology in Netherlands. And he's also the head of the Generative AI group there. And can you pull up the slide, please? Yeah. And today, I'm sure that you'll, before we pull up the slide, I just want to just say that if you have any question, you can feel free to come up to the speaker, the two microphones here. And okay, so now we have the slides available, right? And is this your?
Not yet.
Not yet. Okay.
Right.
Yeah. So after this, we'll have a poster session and networking. We have a reception. You feel free to stay and enjoy networking with your fellow colleagues. I think there's a lot of exciting talk today, kind of span across small molecule drug discovery, and we'll also have some our keynote speaker on protein design, and we will have we are kind of trying to sample across different drug modalities. And and without further ado, I would like to just leave the stage to you. Yeah.
Thank you very much. So first of all, thank you for the invitation. It's great to be here. It's the first time for me in Boston, so it's really a great place. And I am actually a computer scientist. I work on generative modeling / generative AI for over 10 years, so before the hype. I think it's good to mention that these days. And also I happen to be a program chair of NeurIPS this year, so I really liked all the comments in the morning, a bit sour comments. It's actually good to listen to them as well. And I hope that I can also meet you in Vancouver this year, or maybe the next year. And so, as I said, so I'm a computer scientist. I'm not a biologist, I'm not a chemist, I'm not a physicist. So you can very easily kill me with any simple question probably, but I will try to do my best to tell you a little bit about how we can use generative AI to accelerate drug discovery. So a little bit different perspective. So we listen to many different aspects already today. And I will try to point out a few other interesting applications in my opinion. Right. So first I would like to say a few words that you probably know about it, but it's good to always start with why, right? And first why is very shameful self-advertisement. So I just finished my second edition of my book on deep generative modeling. And for me, Gen AI is not equivalent to LLMs. It's really about probabilistic modeling. So I really loved how Jennifer said in the morning that it's about densities, about probability distributions, because this is exactly what generative AI, generative modeling is, right? And we talk about Gen AI, it's actually about parameterizing probabilistic models with deep neural networks, right? And we have many different classes of deep generative models / Gen AI models, like what we listened today already, so diffusion models, latent variable models, energy-based models, autoregressive models like LLMs nowadays, right? And also maybe what is not mentioned here is another class of so-called foundation models that are not necessarily probabilistic models, but they can be used to build more sophisticated generative AI systems. And talking about drug discovery, so like focusing on the R&D part, so of course this is always very lengthy, very costly process, right? And from target identification and validation to lead identification and optimization, in vitro studies, and so on, right? So there are many aspects of R&D before even any drug candidate enters clinical trials, right? So where I see a computer scientist, right, where I see that Gen AI can help in this process, so of course, at this R&D especially, right? So probably, I don't need to convince you about it. The hope is that we can shorten all these pretty lengthy processes And as an outcome, we can get something faster as well and cheaper, right? We saw multiple talks today, multiple great success stories, actually, right? But what I want to highlight is that we can use, broadly speaking, Gen AI not only to get, for instance, molecules, but we can also use it at different places. So, for instance, we can use for target identification and validation. We also listened to that today. And also for understanding mechanisms, why something happens, how a given disease evolves. Because generative modeling, we need to remember, is about actually generative processes. We can say that this is, in some sense, how we think our reality is like. We can sample about our reality, right? So what is like, what kind of condition, what is conditioning, what is condition on this, and so on, right? So this is like a generative process. But also, of course, we can use Gen AI for de novo drug design. As I said, we listen to that, maybe some phenotypic profiling, et cetera, et cetera. And so what we can do with Gen AI, again, so we can look at this from different angles. So one, we can use Gen AI for target acquisition, or like, for instance, with AlphaFold or Rosetta. It's good to remember that there is not only AlphaFold, right? So there are also other approaches. We can also use data analysis, so sequencing data, imaging data, network data, simulation data. There are plenty of amazing simulators that can help us with understanding what are the mechanisms under the hood of given disease, for instance. And of course, we can use Gen AI for molecular docking, de novo drug design, but also we can look at some properties. So this is what I will try to cover to some extent today. So for instance, about enzyme kinetics, right? So we can also use Gen AI for that. We can help chemists in wet labs. We can help them with some tools that speed up their work, all right? So this is very interesting. And there are pretty old concepts, actually, but they come back. So very recently, many researchers from Stanford, Chan Zuckerberg Initiative, they published a paper on Archive about virtual cells, organisms. So to create something in silico, like an organism or a cell in silico, such that we can play with without even touching anything in a wet lab, right? So this is, of course, very, very challenging task, because if you look at a cell, it is like extremely complicated system by itself, and yet we have organisms that consist of these cells, so are built of these cells. So this is like crazily complex, right? But so this is very interesting, and where we can use AI, actually, right, in so many different places. So okay, so how we can use Gen AI. So first, I would like to start with one example of understanding regulatory mechanisms of diseases. So this is a project that I'm involved in with a startup in Amsterdam, in the Netherlands. So the idea is that by regulators, we mean some natural compounds that control biochemical reactions, and this regulation results in, let's say, a disease, right? Or some unhealthy state. An example is vitamin B12. Again, what is in this block, so I can't fully comprehend, but for me, what is important is that input is like some deficiency of vitamin B12, and output is anemia, right? So basically the first and the last word here. But there is some complete system that we can actually understand. Biochemists can understand and explain it pretty well. But this is exactly what we mean by regulators. And so we can actually look at diseases from this perspective. Well, maybe perhaps instead of designing a drug, we can first look and we can try to understand why a disease occurs, right? Maybe we can be lucky enough to find that there is some compound that is natural compound that regulate some process, right? And then based on that, we can start optimizing that. And this is the approach that we use for very challenging problem of Alzheimer's. And we used a database of over 220,000 candidates, regulators that are naturally occurring in our bodies. And we used generative AI based on deep learning, so some foundation models. We created a pipeline, in-house pipeline, that allowed us to actually highlight some potential leads. Then, of course, it's not, don't get me wrong, I am a huge enthusiast of AI, but of course, at the end, or in this process, must be human being, human expert, right? So eventually, medicinal chemists looked at this potential candidate and said, yes, we see in this list some potential candidates, right? Then further, it was optimized in a wet lab. But at least this step was definitely much faster than studying literature for these 220,000 potential compounds, and so on, so on. So we can make it more automatic, right? So this is the first example, very interesting in my opinion. Another example is, like, let's say this was the very beginning of the whole pipeline of drug discovery. Now we are entering like this almost like preclinical step, right? So let's say that we have identified some inhibitor or a candidate, drug candidate, and now we need to check different properties, right? So for instance, enzyme kinetics is one of those steps. So like the Michaelis-Menten model, right? So let's say that like you know more than how it works than me, but there is a system of differential equations. This is what I understand at least. And what we have to do, we have to check how enzyme behaves with and without potential inhibitor. We try different concentration levels, right? And this is the most basic approach. This is the A, right? So this is the so-called standard approach. You try different concentrations, you measure it, you make the Michaelis-Menten plot, and then you get your parameters, right? Values, you estimate your parameters. So our question was, can we do it faster by just having one single measurement instead of multiple, like typically 8 or so, right? So then we, of course, don't waste money. For startups, is super important. We don't waste time. Again, for startups, super important, right? And then maybe we can get very quickly some, maybe rough, but at least some estimate, right, of the value. So this is what we use. So I call it like a local model because this is like for one given case, we can use some computational models. So here we use approximate Bayesian computation. So you can use, if we bend the definitions a little bit, Gen AI for that, right? And this approach gives you very quickly some posterior over your parameter value, right, for this enzymatic parameters. So does it work? Well, we applied it to the case of COVID-19. So with this approach, we were able to actually very quickly check something and put a paper on bioRxiv 2 months after the first cases in the Netherlands, right? So it was like around March, and in May we already had some first write-up, right? And this was possible because actually we applied this computational approach. So we really sped up everything, and then the paper was then published 1 year later, right? Because we had to do everything properly, but at least it helped to very quickly navigate to say, hey, it's actually worth to pursue, right? And after all, this is what we do care about, right? We want to quickly proceed. Like, you know, like I always joke that in AI, if a paper is 1 month old, it's pretty old. If it's 1 year old, it's like ages old, right? So I think it's getting like that everywhere, right? in medicinal chemistry, it's getting similar, right? So this is an example that actually we can use that. And very recently, so this is preliminary works. It's unpublished, very preliminary. Again, with enzyme kinetics. And what we try to use here, we try to look at the problem as time series forecasting / generating, right? Time series. And the case is here that we assume that we don't know for a second that there are some ODEs, there is some Michaelis-Menten model. We just have some measurements. So what we can have substrate concentration, enzyme concentration, and then we observe the curve for product, right? How it changes over time, right? And we can phrase it in two ways. So first, we can say that, hey, we have some initial measurements, and we stop it, right? And we say, now, given those data, this and this concentration, initial concentrations, predict the remaining of this curve, right? It could be useful, but typically, we are more interested in another scenario that we only have the initial condition for the product, which is 0, and we don't do any measurements in the wet lab, and we want to generate or predict the remaining of this time series, right? And for our initial experiments, we use Michaelis-Menten model to generate like synthetic data. But of course, in practice, we can say that, hey, we are now agnostic to this enzyme kinetics model under the hood. It could be anything, just provide me a lot of data. Also, if this works, it has this very nice property that since this is a deep learning-based model, we can now add many other modalities, right? So with ODEs, we need to stick to ODEs. With deep learning models, we can attach many different components and as I said, modalities. So for instance, we can now give structures of enzyme and ligand, and we can train such a model, right? So, but first comes first. So first, we wanted to check if even this neural network-based approach works. And in the first case, we were pretty positive that it will work. In the second case, we were something like, probably it won't work, right? I work with my PhD student, and as a good supervisor, I said, it probably won't work, but let's give it a try anyway, right? And additionally, so we wanted to check if giving these initial conditions gives some information. It should, because this is how the ODE works, right? But we wanted to check it as well. We picked like state-of-the-art model. Again, state-of-the-art in terms of AI, it's a bit tricky. So in April, they were state-of-the-art, not anymore, but they were pretty good. And so these are some results. By the way, I hate looking at tables, but this is the only table here, so bear with me. So if this scenario 1 is this, let's say we have more information, and scenario 2 is that we don't give any information of previous measurements, right? And so if we don't use any external information, so we only give no initial conditions are given, so the model performs extremely bad, which is a very good information for us. Because when we give this information, initial conditions, suddenly everything works pretty well, right? And here is some very cherry-picked example. Yeah, sorry, so this is actually scenario 1. I mixed them up. So this is scenario 1. So if you give some initial information, it is capable of predicting everything very well. If you don't give this external information, the prediction is pretty bad. And then, as I said, so this is unpublished, so unfortunately, I don't want to share too many information with you, sorry, guys. But if now we updated the model in some way, and first of all, it worked better in this scenario 1, but also worked better in this scenario 2. And this is actually very, very interesting and very promising. And this is actually how, like, I think that a very skilled specialist expert would work. So how it works, if you give initial conditions, and if you know a bit how this ODE works, so the initial value of the substrate will be the upper bound for the maximum of the product, right? Because of, well, you cannot get more product than your initial substrate, right? So then what we notice is that exactly by giving this information, so you can see that this green and red, so these two curves, so they exactly like go to the maximum, right? If you don't give this initial condition for a substrate, the model goes completely bananas, right? It goes somewhere. And we noticed that by giving this external information, so this model worked a little bit like that. It figured out the curve, so it was always something like this logarithmic, let's say, right? And then by giving these conditions, it was just shifting it up or down. And then when we discussed that, we realized that probably this is how we would work. Let's say you are given 1,000 examples. You see that all these curves are a little bit like that. And then by having different initial conditions, you just move them up or down. So you have something like an average of your curve, and then you're moving up and down. And this is actually very cool, because now maybe if we give extra information, like for instance, what is the structure of your enzyme, what is the structure of your compound, maybe the model will start figuring out the dynamics, like the rate, right? So maybe it will start moving left or right. So this is our stage at this point. So we are working on that. I think it's super, super interesting. And it can, like, at the end of the day, if we can create this, let's say, foundation/GenAI model that you don't need to run in a wet lab, you can run on your laptop, it's like amazing, right? Because you can very quickly verify for given enzyme and compound what is the enzyme kinetics of that. Right. And the last example, sorry, when I'm getting excited, I don't know how much time do I have. I should finish probably. Yeah. All right, sorry. So very quickly, so this is about mechanism of action. So phenotypic profiling with images. So we used a data set that was actually prepared here in Broad Institute. And this is an example of like a follow-up paper of a platform or framework that we developed earlier in Amsterdam, at the University of Amsterdam, on attention-based multi-instance learning. For histopathology slide, we use it for other data. And we used like pre-trained embedding models, so like a foundation model, on top of which we created a Bayesian model, right? Not going into the details. What was interesting is that, oh, sorry, another table. I lied to you. So there are two tables in this presentation, another table. So we have, obviously, sorry guys, like AI spirit in me. So we got state of the art, obviously. But what was interesting is that because it has this attention mechanism, for everyone who is familiar with attention mechanism, so you can actually use attention mechanism to highlight which parts of the input are important or not, so where the model put some emphasis on. And our model, this is, it looks like cherry-picked. It was, but in general, it worked pretty the same way. So you can see that actually our model put a lot of emphasis on these small images with some cells inside, and put very low for the background, basically. So this is, again, how you would expect this model to work, right? So really, really interesting part. And so to conclude, I don't want to go through that, but in my opinion, this is very interesting how we can use GenAI for really different applications, not necessarily only molecule generation, but really in many, many other parts of the pipeline. And personally, I believe that this virtual cells organism is like probably the way to go. I would like to thank everyone, and if you are interested, we can talk more. And in a month, I'm moving to San Francisco. I'm joining Chan Zuckerberg initiative, so maybe we can also collaborate in this continent. Thanks.
All right, thank you so much. Given the time, maybe can we have one question? If you have, can you come to the stage? While maybe can you, the AV team, can you set up, put Jen and the Zoom on the screen? Thank you.
Hi.
Go ahead.
Hi.
Was it? Yeah. Hi, my name is Nozay Ahmadi from Brown University. I don't know much about like enzyme kinetics, but
Yes.
I've seen your model. I don't know why you call it generative AI, because it's more like neural operator or like the foundation model.
Right.
Yeah, but I have a question that what is the input of your model that you get the kinetics?
Right. So thank you. Very good question. So indeed, it's a little bit how you look at the problem. So like in general, time series forecasting is a perfect example of an autoregressive model. And an autoregressive model is a generative model. But I'm not so like, you know, to me it's tomato a bit. So you can call it whatever you want, right?
Okay,
But what is different here is, because I saw your poster, by the way,
yes.
so very cool one. So it's, we assume that we don't know what is the ODE. So we don't use these neural ODE kind of ideas.
Yes.
We really want to be like fully data,
Yes, but
data
what is
centric.
the input of your model?
Yes,
Is
so
it like the
like initial conditions and maybe some
parameters
part
of the model?
No.
Only
We
data.
don't know. We don't know the parameters.
Okay.
That's why it's not working so well, maybe, right? But we have only the initial conditions and part of your time series, and that's it.
Okay.
So you don't know what is under the hood.
I see.
Of course, if we know it, and we should use it. So this is, but this is for our future work.
Yeah, thank you so much. I
Thank
have
you.
a similar work on like the effect of the drug on tumor growth. So we have state-space model Mamba for
Right.
predicting the efficacy. So in that case, we have like more feature and not initial condition at all, but like more feature about the patient. And
Nice,
so on.
very
Yeah.
cool. I will come back.
Very
to
interesting
your,
talk.
yeah.
Thank you.
Thank you. Thank you very
Thank
much.
you. Okay, yeah. Let's
Yeah,
give
thank
the
you.
speaker one more round of applause.